Loading samples into the EMTP begins with the loading plate. This allows putting samples into the basket assembly. The stem, this metal pin, will hold the samples throughout processing. The samples are held in two types of baskets. This is a large basket. It has 3 wells, a notch for alignment, and the center hole is where it fits onto the stem. The loading plate allows baskets to be placed with reagent as needed to keep the samples wet. There are 2 rows, one for large baskets and one for small. A small basket has more individual slots for samples, but otherwise functions in a similar fashion as the large baskets. 3 large and 1 small basket can fit, or 7 small baskets. Loading up an assembly includes taking a small metal pin and placing it in a slot appropriate for the number of baskets. 2 slots for a large basket and 1 slot for a small basket. Next is a lid to prevent samples from falling out the top of the basket. Each basket is loaded simply by pressing the stem down through the center column of that basket. The lid keeps samples in at the top basket, whereas each lower basket is sealed by the basket above it. The last step is attaching the foot. This screws into place and prevents the baskets from separating. Take care to tighten the foot onto the stem fully so that no gaps can appear between the baskets or the lid at the top. Once a basket assembly is complete, it can be stored in a reagent container to keep the samples wet as appropriate. After processing is complete, the basket assembly is taken apart in the reverse order of the prior steps. First, the foot is unscrewed. Each basket can be removed by just gripping it gently and slowly rotating the stem to pull it loose. The notch in each basket ensures it can be lined up to keep track of where the samples were originally. During the separation of the baskets from the stem, be careful not to tilt the baskets that would dump the samples out. The lid of the baskets can be set aside as it is no longer needed after disassembly. An important part of the cleanup is cleaning the stem, the pin, and the foot. These metal components should be washed in an appropriate solvent, usually acetone or ethanol.